My name is Ronald Dijkman. I'm a virologist at the Institute for Infectious Diseases here at the University of Bern. My group is interested in emerging viruses, and I think we all know emerging viruses by now with the example of SARS-2. And we're trying to find out or figure out which factors contribute that a virus can emerge from an animal population into a human population. And by doing so, we're trying to study how those viruses interact with the human tissue, but also with the animal tissue. So to study SARS-2, why it's different from its little sister SARS that appeared early 2000s, we use primary airway epithelial cells that we isolate from human patients and that we then reconstitute in vitro in a transwell format where we can then study those viruses in more fundamental details. Our research showed that this SARS-2 virus replicates much better in the upper respiratory tract, and that might explain why this virus is so widespread as we know to date. But by using this model, also all the subsequent research that we did in collaboration with other researchers, this model was one of the pillars to characterize the new variants, what their impact is. I think the biggest challenge is knowledge, that there are models that can be used to just answer fundamental questions, so you not per se need to use an animal model or a mouse or another animal. But I think also one of the biggest bottlenecks that we now see is the costs. The cost of chemically defined reagents is much higher than doing the same experiment in an animal model. So in our line of research, the model that we used, and that was also recognized by the 3Rs for the awards, is one of the main pillars in our research. And we continuously expanding this to other species. And we also would like to share our knowledge, but also the models that are the availability to other researchers to lower the thresholds of using those models. If I had time, I would create a non-profitable organization that other researchers can contact and that we then disseminate the knowledge, but also the reagents to implement these type of culture models in other people's labs.